Assalamualaikum warahmatullahi wabarakatuh. My name is Nur Fitria binti Ahmad Munir and my matrix number is 23DBT 20F1036. Today I want to show you how I perform a virtual lab about bacterial identification. So the purpose of the lab is to familiarize with the science and techniques used to identify different types of bacteria based on their DNA sequence. Not long ago, DNA sequencing was a time-consuming, tedious process. With the readily available commercial equipment and kits, it is now routine. The techniques used in this lab are applicable in a wide variety of settings, including scientific research and forensic labs. So there are the basic steps for this virtual lab. First, prepare a sample from a patient and isolate whole bacterial DNA. Second, make many copies of the desired piece of DNA. Third, sequence the DNA. Fourth, analyze the sequence and identify the bacteria. So we can proceed the virtual lab. This lab have six samples, which is sample A, B, C, D, E, and F. Sample A is fluid from lymph node. Sample B is stool sample. Sample C is urine sample. Sample D blood sample. Sample E is sputum sample. And lastly, sample F is stool sample. So for part 1, sample preparation. So first, please put on a pair of rubber gloves by clicking the glove box in the open drawer. Now, obtain a sample from the culture dish. Click the wire loop to continue. Click one of the bacterial colony in the dish. Okay. Next, click the microcentrifuge tube to transfer the bacterial colony from the wire loop into the microcentrifuge tube. Placing bacterial. Now add digestive enzymes to sample. Click on the yellow micropipette to continue. Adding the digestive buffer to the microcentrifuge tube. Discarding the tip for prevent contamination. So, the microcentrifuge tube must sit for several hours. Click the microcentrifuge tube to continue. Okay, waiting for several hours. Okay, now after several hours of enzymatic digestion, sample is ready and click the microcentrifuge tube to heat inactivate the digestive enzymes. Moving the microcentrifuge tube to the water bath for heating. Next, the digestive enzymes are now denatured and inactivate. Click the microcentrifuge tube to continue. Okay. Now Now spin down cellular debris for removal from sample. Click the centrifuge lid to continue. Okay, centrifugation is complete and unwanted debris have now been spun down. DNA we want is in the supernatant. Click the microcentrifuge tube to continue. Now transferring the supernatant to the PCR tube. The sample preparation is complete. Click the orange red to continue to part 2, which is PCR amplification. So we proceed with PCR amplification. Now add the PCR master mix solution to all tubes. Click the red cap master mix bottle to continue. Adding the master mix solution to the PCR tubes. Now set up the positive control reaction. Click the green cap positive control DNA bottle to continue. Adding control DNA to the positive control PCR reaction tube. Okay, for the negative control reaction, add water. Click the blue cap deionized water bottle to continue. Adding water to the negative control PCR reaction tube. Click the PCR machine to open the loading compartment and load the PCR tubes. So the PCR tubes have successfully been loaded into the PCR machine. Click the PCR machine to continue. So running PCR amplification, click above to pause or replay the animation. Click done when finished. So wait for the running PCR amplification. So finish. Click done. Now the PCR amplification is complete. Click the orange tray to continue to the PCR product purification process. So for the PCR purification, first set up the micro concentrator column. Click the yellow cap buffer bottle to continue. Adding buffer solution to the column. Discarding the tip to prevent contamination. Click the orange tray to continue. Adding PCR product to the column. Discarding the tip as usual to prevent contamination. So next, the tubes containing the PCR sample, which is positive control and negative control, will be put on ice. Now click the orange tray to continue. Putting the tubes on ice. Click the orange tray to load the column into the centrifuge. Okay, loading the column into the centrifuge. Adding the counterbalance to balance. So next, click the centrifuge to continue. Wait for 15 minutes and unloading the centrifuge. Now DNA is trapped in the column. Now loosen it into a new tube and click the orange tray to continue. Adding a new collection tube, inverting the column and click the inverted column to continue. Adding buffer solution to the inverted column. Then discarding the tip. Okay, next, the first collection tube is no longer needed. So click the orange tray to discard it. Now, centrifuge the assembly to collect the DNA into the collection tube. Click on the orange tray to continue. Loading the column into the centrifuge. Then adding a counterbalance to balance the microcentrifuge and click the centrifuge to continue. Centrifuging for 2 minutes and unloading the centrifuge. Now, DNA has now been collected in the collection tube. The column is no longer needed and will be discarded. Now click the orange tray, discarding the column. Okay, next, the PCR product has been purified. So, click the orange tray to continue to part 4, which is DNA sequencing. So, for the sequencing preparation, first dilute the PCR product to increase the volume. Click the blue cap distilled water bottle to continue. Adding distilled water to the purified PCR product and discarding the tip as usual to prevent contamination. So now the green and blue strips tubes already contain PCR reaction mixtures in them. So now clip the strip tubes to continue. Adding purified PCR products to the strip tubes. So one by one. Okay, done. Okay, the strip tubes are ready for PCR amplification. Click the PCR machine to continue. Loading the strip tubes. And the strip tubes have been loaded. Click the PCR machine to continue. Running PCR amplification. So wait for the animation. So done. So the sequencing preparation is complete. Click the orange tray to continue to part 5, which is DNA sequencing. So for DNA sequencing, now load the automatic sequencer. Click the orange tray to continue and click the DNA sequencing machine to open the doors and load the tray. So clicks the orange tray to load it into the DNA sequencing machine. Loading the tray. Now click the inner green door above the orange tray to view the DNA sequencing animation. Now sequencing and wait for minutes to see the animation. Okay, done. So click done. Now DNA sequencing sections complete. Click the orange tray to continue to part 6, which is last part, which is sequencing analysis. Okay, for the sequencing analysis, follow the instruction in the notebook window. Click the computer screen when you are ready to identify the bacteria. So, for sample 1, identify the bacteria. Sample 1 is sample A. Sample A is Bartonella Hensley. Okay, answer recorded. Click the samples tab to perform DNA sequence analysis on other sample. So for sample B is Escherichia coli. Okay, next. Sample C. The bacteria of the sample C is Pseudomonas aeruginosa. Okay. For the next sample, which is sample D. Sample D is Salmonella typhimurium. Okay, for the next sample is sample E. Sample E, the bacteria is Yersinia pestis. So, next. For the last sample, which is sample F, so this bacteria is Yersinia enterocolitica. Okay, it's done. So that's all for this virtual lab. So thank you.